Hello, everyone. I am Wen Shen. I'm an assistant professor at the Mechanical Aerospace Engineering, as well as, I'm also affiliated with the Nanoscience Technology Center at the UCF. So today I would like to talk about my research about developing microsensors for biomedical applications. So here is a general overview of my lab. I'm focusing on the functional materials and devices development. So there are a few research thrusts currently in my lab. Biomimetic devices for human or animal physiological interfacing, wearable electronics for biosensing, drug delivery, embedded sensors for structural health monitoring, which is a little bit different, but and then biosensors for agricultural and environmental monitoring. These are a few examples of our previous research. First of all, in today's talk, I would still like to give you an example of this implantable sensor that we have recently developed. So this sensor is actually a brain neural interface. And then we all know that the neural interface electrode can be used for scientific study, for medical diagnostics application, as well as for brain-machine interfaces. And then a big challenge in the device development right now is that the current devices are not very compatible, are still not compatible enough with the human tissue. The reason being that the materials are foreign to the neuron tissue. So there is always a lot of the active reaction occurring at the implantation site, forming basically a scar tissue around the implantation site, and then these scarring tissue is impeding the signal to noise ratio of the signal transmission. In addition to that, these scarring tissue are not very good for the tissue itself as well. So the approach that we are taking to solve this challenge is that we're looking at how we can make these neural interface devices using more natural materials. So the material that we have chosen is called extracellular matrix materials. These are materials that are secreted by cells, and then these materials form the structural support of the neuron tissue itself. So you can see that these are some major components of the ECM materials. These include these long fibrous structure of collagens, as well as fibronectin, laminin, and some small molecules that give different biological functions in the tissue. So then, as an engineer that develop these devices, we are looking at how we can fabricate these biological materials into microelectronic devices. So in my research, I've been focusing on, first of all, the fabrication of microelectronic devices using these more natural materials, ECM materials. So you can see here is one example of microfabricated ECM-based neural electrode. And on the top line is the microfabricated silicon-based neural electrodes. You can see that our fabricated ECM electrodes have the similar size and geometry as the silicon microelectrodes. However, the materials and the surface structures of these ECM microelectrodes are significantly different. You can see these long fibrous structures of collagen still remaining on these fabricated ECM materials. And we believe that these structures will support more bio-friendly environment after implantation. So here is a fabrication process that we developed. This is just one example that we are using micromagnets. Basically, they're magnetic particles in a very small microscale as a contact mold to form the small micro-sized geometry for these bio-ECM materials. And then we use multiple contact transferring methods to encapsulate soft, flexible electronic devices into these ECM substrates, and then we align them into some prefabricated stents, and then use these stents as a laser mask to final ablate the outer shape of these electrodes. So after fabrication, we have collaborated with our collaborator to check on the biocompatibility of these electrodes. We found that neuron cells can grow very healthily on top of these ECM bioelectrodes directly versus flexible electrodes that does not contain these ECM materials. Neurons cannot grow very well on those non-biological electrodes. And then we also implanted these electrodes in the animal. We found that the conventional electrodes that are non-biological material made will form a scar around the implantation site, but for ECM biomaterials, we don't see significant scarring around the implantation site. One extra step we also did was we also implanted these bio-ECM electrodes in the animal for a much longer time. So these results are after 1 month, but then we have also followed up the experiment and implanted these electrodes in the animal, in the rats for over a year. And then we still see very good biological responses and also neural signal recording responses. So those new results will be, we are planning to publish that soon. And then in addition to the biocompatibility benefit, we also looked at the mechanical property of these bioelectrodes. So these basically, these materials are a type of hydrogel. So they are very stiff when they're dehydrated in the air. So that will facilitate the implantation of these electrodes. But after implantation, then the fluid in the tissue will hydrate these hydrogel, and then they become very soft. So you can see the Young's modulus has decreased 1,000 times after hydration. And then, as I mentioned, we also performed neural recording. We found that these electrodes can record local field potential. They can record the spiked band frequency and multi-unit activities. So we have also evaluated their neural recording performance after these devices has been implanted for a year, and they so far they looks very good. In addition to the neural electrode application, we have used the similar fabrication and also designing we have also used the similar fabrication technology to design other wearable sensors and also other hydrogel-based sensors for detecting drugs as well as for cell interfaces. So hopefully I can show you some new results soon. And then I think I have a little more time. I would like to focus to also include another different study where we use the magnetic materials to produce sensors. So here is an example of making sensors using a giant magnetostrictive materials. So by designing these sensors, we're able to make very small thin film sensors that are in the micrometer size. So the largest dimension of these sensors are 500 micrometers in size. And then these sensors, when we place them on top of the target. So here we use the tomato as an example. We spiked some salmonella contamination on these tomatoes, and then if we can place these micro-sized sensors on top of these tomato surface, they can pick up the target pathogen, which is salmonella here, and then the increase in weight of these target pathogen will cause the resonant frequency of this sensor to decrease. So we can see that we can measure in real time the decrease of resonant frequency caused by the detection of the target pathogen. And actually, by using the similar technology, we're designing other type of sensors, for example, for structural health monitoring, and also for other detections of targets in near field. And then we're also designing the detection system, including the antenna and also the entire detection system, so that we are trying to develop, we're trying to detect a target from tens of meters away in the field. So this is another example of the sensors that we're developing in the lab. And then finally, as a summary, in my lab, we're developing biomimetic sensors for physiological interfacing and for agricultural environmental monitoring and for some other applications such as structural health monitoring. So I'm also looking for other collaborations in these fields. So that's the end of my presentation.